Hello, hello everybody. Can you hear me? Can you hear me? Sorry I was late, people. My computer died on me all of a sudden. I'm not sure what the problem is. So I had to rush and get my wife's. So my camera's better today, basically. You can see how bad my hair is right now because my camera's better. Um, Lake, don't laugh. Your hair's a mess too.
Yeah, it always is.
All right, so we're starting a little late, so let's just go. Oh, wait, before we can start, I need to go to Google and get my files. So hang on. Hang on, hang on. Right. Working on it. What do you want? Cells download. What? What do you want, kitty? Almost there. My wife's computer is really slow, but it's better than dead, which mine is. Sorry. Got the wrong file downloaded. Oops. And I have my phone. What else could a man possibly ask for? Okay, sorry about the delay, people. We are back on track. 15 minutes late, but you know what? We always end up 15 minutes short. So what are you going to do? So today we're going to start chapter 24. We have 3 lectures on chapter 24. It's about the cell cycle. And it's in freshman biology. And I tend not to do too much info on mitosis itself because it's really bio 101. I don't have a lot to add to it. After we get through a little ways, we will see some of the details inside mitosis. But for the most part, you know, chromosomes condense and they get segregated. So we won't spend too much time on that. But that's where we start. Oh, no, I'm sorry. We'll start with the cell cycle itself. Oops. Who needs notes? I'm going to wing it today, people. This is the 28th year I've given this lecture. In some years, it's 2 or 3 times in the year. So maybe I can just take it off the top of my head. We'll see. All right, so cell cycle. And this is for eukaryotic cells. They go through this cycle of different phases in their division. This whole cycle is, on average, depends on the cell, depends on a lot of things, but let's say about 24 hours long. And if the whole cell cycle is 24 hours, the actual mitosis part of it is only 30 minutes. So when we talk about a cell going into the cell cycle, usually we say it's going through mitosis, but mitosis is a really small part of the whole deal. So mitosis is really all about the division of the chromosomes. The cell itself doesn't split in mitosis. Mitosis is all about condensing the chromosomes, segregating them, so a half goes to each side of the cell. And then the next thing is meiosis, which is just this straight line here, which is when the cell actually splits into two cells. So when that happens, you've got a cell that is maybe a little bigger than a normal cell. It splits in two. Now you've got two cells that are really, really small. So, well, really, really small. They're probably 3/4 size. So after the cell divides after mitosis, it goes through G1, where the cell goes back to a normal interphase type cell, and it grows up to a normal cell size. So that's G1. Let's see, can I get a pointer option? Highlighter, nope, laser pointer. Meow. Okay, so now we're in G1. So as the cell gets towards the end of G1, it has reached its normal adult size and it's happy. Now it could stop there. We'll talk about that shortly. But if it's going to stay in the cell cycle, the next step is to go into S phase. And S is called S for synthesis. Here we've got replication going on and the genome is getting duplicated. So the cell is going from 2N up to 4N, has 4 of every chromosome. And then after S phase, it goes into G2. And now G2 is like, shortly we're going to split into 2 cells and each of them would be half as big as they need to be. So G2 is a growth period where the cell gets bigger, makes more organelles, makes more of everything it's going to need when it becomes 2 separate cells. So you can kind of think of it as going from a 1x size up to a 1.5x size. And then after mitosis, you have 2.75 cells. And then they grow again in G2. So, or G1. So G2 is basically growing up to a little bit bigger. And also there are a lot of checks here to make sure that the chromosomes have been totally replicated and that, whenever you do replication, you have DNA errors that creep in. So all those errors have been fixed. So all the checks to make sure the cells are ready to go into mitosis happen here in G2. And then you go into mitosis and you split into 2 cells. So that's the cell cycle. Here is mitosis up close and personal. Our microtubules from earlier, I said that they start near the nucleus here, between the nucleus or between the rough ER and the Golgi or something. So here's our interphase microtubules, and that all starts at an MTOC. That MTOC then duplicates itself, so you have 2 MTOCs. And now we're going to go into prophase. So prophase is often split into early prophase and late prophase. And sometimes they call it prophase and prometaphase and then metaphase, but we're going to call it early and late prophase. So early in prophase, 3 things are happening. 3, count them, 3. One is you're going to get the disassembly of the interphase mitotic spindle, and then, interphase microtubules, and have them, and they're going to start reorganizing into the mitotic spindle. Hang on, I'm trying to do something here. Okay, so let me just check. Oh shit! Oh, sorry guys, my PowerPoint just went out on me. Heck. I'm having a bad day. Will you forgive me if I whine a little bit? I'm having a bad day. Okay, so prophase, 3 things happening here. One is the cytoskeleton's rearranging. Oh, let me share again, sorry. Claire's getting sick of my shit. I can see it. I can see it in your eyes, Claire. You're tired of me already. Okay, so three things are happening here. One is that cytoskeleton is rearranging into the mitotic spindle, and that involves the, once the MTOCs have duplicated, they become asters. And these asters are going to move towards the poles of the cell. Another thing that's happening between here and here is that the mitotic or cytoskeleton's coming apart to do this. So the cell's going from whatever shape it is into a round shape. And that's going to help with the division as well. So the whole shape of the cell is going to change here. Another thing that's happening is the nuclear membrane is starting to break down. Now in a light microscope, it looks like it just disappears, it goes away. What's actually happening is the proteins on the inside of the membrane that are holding it, keeping it intact, are being phosphorylated. Those are the nuclear lamins. I talked about them in chapter 13. Remember, we talked about microfilaments, microtubules, and intermediary filaments. This is one of the only intermediary filaments that I talked about. And that's because it comes into play here. It's really important. So this is an IF. It coats the inside of the nuclear envelope, and it will get phosphorylated and depolymerize. When it depolymerizes, that nuclear membrane, a double membrane, is going to fall apart into vesicles. So it's not disappearing, it's just not visible in the light microscope because it's really small vesicles at this point. And then the third thing that's happening is the chromosomes are condensing. Again, in terms of visibility, in interphase, you can't see chromosomes at all. Here's a picture where you've stained the DNA blue, and it just looks like a diffuse blue color throughout the nucleus. You can't pick out individual chromosomes. Here in early prophase, you can see them starting to distinguish themselves. Then late prophase, basically all these things are just completing themselves. They will finish off. So the chromosomes will totally condense, so they become those X-like structures you see in a karyotype. The microtubules have, asters have reached the poles, and the mitotic spindle is formed, and the mitotic spindle now is attaching to the center of the chromosomes. A little bit of fragments of the nuclear membrane left, but that will eventually just become all vesicles. So these microtubules are then attaching to the center of that chromosome, which is a bunch of proteins in the center that have accumulated on the center called a kinetochore. We'll talk more about that later, but our microtubules now are attached to that kinetochore, and they're going to start pulling on those chromosomes. So then we get to metaphase, where the chromosomes all line up at the center of the cell at this theoretical line called the metaphase plate. The metaphysical plate, no, I made that up, metaphase plate. So that's metaphase, and the cell is going to pause here for a second. We'll talk about that checkpoint later. So keep in mind, once they all line up, the cell stops for a couple of minutes, and then we go into anaphase. Now, two things are happening in anaphase. One is that kinetochore breaks apart and the two sister chromatids separate. And they start to go towards the opposite pole. The other thing is, notice the cell is elongating now. So over here, it was a circle. Now it's an oval. So the mitotic spindle is pushing against itself and stretching the cell out. And then once those chromosomes reach the poles, we hit telophase. So telophase is the opposite of prophase. So everything that happened in prophase unhappens here. The chromosomes decondense and they become diffuse again. The lamins get dephosphorylated, they repolymerize the nuclear envelope, and the mitotic spindle depolymerizes and then becomes the interphase spindle again. one thing I forgot to say up here, let me just back up a tad. Protein I want to throw in here because we talk about it later. So here, this condensation of the chromosomes from diffuse into packed up happens because of the phosphorylation of a protein called condensin. Okay, so we'll see that come into play again later when we talk about getting through the G2 to M transition. So condensin gets phosphorylated, that causes that to happen. And nuclear lamins get phosphorylated, which causes the nuclear membrane to go away. Mitosis. So chromosomes condense, they line up, they separate. Here's metaphase plate here. Why does it say prometaphase, metaphase? This looks like interphase, prophase, prometaphase, and now this is metaphase because they're lined up here. I think that's labeled incorrectly. And then here's anaphase and telophase. Okay, again, that's Bio 101, but now we'll see a little more detail on it in the next 15 minutes or so. And then that last step, the separating of the cell into two cells, is called cytokinesis. So in cytokinesis, you get a cleavage furrow coming down between the two cells, and that's what we were looking at with Xenopus early development too, that cleavage furrow. So in animal cells, with no, and fungi as well that have no cell wall, this cytokinesis is caused by a contractile ring of actin and myosin. So that's microfilaments that are anti-parallel and they're overlapped by myosins, and the myosins push them like this. And it's just like a belt tightening down, tightening down, tightening down till it gets so small that it pinches them off and they become two separate cells. So that's cytokinesis in animal and fungi. In plants, it's different. Plants always have to be different, don't they? So plants are a little different. They have a cell wall, so just making a plasma membrane isn't going to cut it. So what happens is you get vesicles coming that have cell wall material, and they fuse between the two nuclei, and they form this cell plate, and then the cell plate becomes a cell wall between the two. And of course, there's plasma membrane on the inside of both sides of this cell wall. So plants. If you want to know anything more about plants, you got to talk to Dr. Burgess, because I'm kind of stupid when it comes to plants. Okay, so that is cytokinesis. So now, like I said, a little more detail on what's going on in terms of the proteins involved in all these movements that are going on. Look at my notes real quick. So role of microtubules in this. And by the way, in the notes, I've got a bunch of videos embedded in them. I would like to show them now, but I already killed 15 minutes of the class by having computer problems. So you should look at them yourself when you get a chance. But we've got 3 different types of microtubules involved in this. Of course, they're all structurally the same. The only difference is what their function is. So one type of microtubule is the kinetochore microtubules. Okay, so this is all like here and here. And here you can see 3 different, come down to a better one here, 3 different types of microtubules. One are the kinetochore microtubules that attach to the chromosomes, or sister chromatids, I should say. Each half of this is a sister chromatid. Then you've got the astromicrotubules, and they are going from the centrosome out to the cell wall, cell membrane, and they're attaching to the cytoskeleton at the cell membrane. And that's anchoring the centrosome here at the side of the cell. And then you've got these microtubules that are polar. They go from one pole of the cell to the other pole of the cell, and they overlap parallel kind of a manner here. And between those polar microtubules, they're overlapped by molecular motors. And these are motors that are going to walk towards the positive side. So these are kinesin motors here. So you've got kinesin here and kinesin here. Each of them are moving in that direction. So let's look at the kinetochore a little closer. So here is a microtubule, or I'm sorry, here is a chromosome in blue, and then here are microtubules attaching to that kinetochore. Here, protein there and there, all around the center, and these are microtubules coming off of it. So what's happening here is your microtubules, as they're growing out, polymerizing out from the centrosome, they will eventually run into the outer core of the kinetochore. And this kinetochore has got this fibrous corona. So what happens is the microtubule comes in and it hits here, and then the fibers of that corona then will attach to the microtubule, and they'll stop it from depolymerizing. So it's going to do a microtubule capture here. So now the microtubule is attached to that kinetochore. Okay, so and this is really important because, we're just showing you a little bit of what's going on here. You've got your microtubule and these connecting proteins from the corona, but there are other proteins there as well. And when the microtubule comes and connects to that corona, they displace these other proteins. They're called MAD and BUB proteins. So later when we talk about the metaphase checkpoint, when the chromosomes pause there in the middle of metaphase, it'll be involved with these proteins that are coming off the kinetochore when microtubules attach. So we'll talk about that more later. Little preview of coming attractions. Okay, so back to our centrosome. You've got a molecular motor here that's walking from plus to minus. So that's a kinesin here that's pulling the microtubule towards itself. And as it does, the microtubule goes past the motor and then depolymerizes and comes off, because these polar microtubules are going to be getting shorter and shorter. Here in the kinetochore, you also have molecular motors. These are going to be dynein, and they are walking towards the minus side and pulling the chromosome with it. So you've got these chromosomes moving based on motors at both ends. And depolymerization as it goes past. Now with the astral over here, you've got molecular motors, dynein here attached to the cortex, grabbing onto these microtubules and pulling them in this direction. Okay, and that's kind of holding that that centrosome where it is. Otherwise, when the kinetochore pulls it, it might pull the centrosome instead of pulling the kinetochore. So we've got attachment from the cell wall to the centrosome out to the kinetochore and pulling those microtubules in. And then last but not least, your polar microtubules here are overlapping, and you've got this motor in the middle, again, a kinesin moving towards the plus side. And as it moves to the plus side, this microtubule is actually polymerizing and getting longer. So this one's getting shorter. This one is possibly getting shorter. These are getting longer, and that's forcing the poles of the cell out and stretching the cell out, getting ready for cytokinesis. So those are three different types of motor proteins. Questions, comments? All right, let me check my notes real quick to make sure I haven't forgotten something important. Okay. All right, so that's the basics of mitosis. So now we get into what determines whether the cell is going to go into the cell cycle or not. Is it actually going to start dividing, or is it going to stay as a non-dividing cell? And that has to do with these cell cycle checkpoints. All right, so we've got two main checkpoints in the cell, okay, and then one more minor one over here. So this minor one is very different from the other two. The molecular mechanism it takes to get through this checkpoint is very different from the other two. But these two are very similar. They're controlled by similar mechanisms. Now, this one is the most important one. We call this the restriction point, or the R point. And that's because most cells are going to be out here in G0, or at the very least in G1, and then pause at this point. Getting through this point and into S commits the cell to divide, because once it goes into S phase, it replicates its DNA, and a cell can't survive with that many chromosomes. So it's either going to complete mitosis or it's going to go into apoptosis here. So this is an important control point that decides whether the cell is going to go ahead and go through the cell cycle. Once it does, well, this is the only point where the cell is responsive to external factors, like growth factors. Growth factors will tell that cell to divide, but it gets it through this point, and then it has nothing to do because it can't affect the cell anymore. Once it goes into S phase, there's an internal clock that keeps it progressing forward until it comes all the way back to here, and now it's responsive to external factors again. So that's why at this point, if a cell loses control at this point, it just keeps dividing and dividing and dividing, and that's cancer, right? So controlling this point is also really important to controlling the cell so it doesn't become, go out of control and become cancerous. So this is our key control right here. Now, most cells are going to be over here in G0. Most of the cells in your body are in G0 right now. They're not dividing. They don't have to divide. They're adults, they've differentiated, and they're performing some function. So they're just going to be over here, and stay over here, right? Like your nerve cells, for the most part, are never going to divide, your muscle cells too. So they're just sitting over here. But some cells can be induced to start dividing again. Things like liver cells. If you do damage to your liver, then your liver can repair itself by releasing growth factors, and then the good cells divide, and then you get more liver cells. So some cells are staying in here. Some cells are mostly here, but can be induced to go back in the cell cycle. And some cells are just always in the cell cycle. Your stem cells are constantly dividing because you constantly need new cells to replace ones that are dying. And here we're talking about hematopoietic stem cells that are making more blood cells, skin or epidermis cells that are constantly dividing. So those are ones that are epithelial, replacing the skin that keeps sloughing off you, and also intestinal, right? Those are constantly dividing to basically clean your intestines out, get rid of any toxins. Like the inside of your intestine, you consider, I don't know if anyone else feels this way, but it's pretty friggin' gross as far as I'm concerned. You got a lot of toxins and crap in there. So if you let that build up in cells, they'll mutate and become cancerous. So they're constantly sloughing off and being replaced with fresh cells. So your stem cells then are constantly in this cycle and making more and more cells. So this is your restriction point where all of that is controlled. Now over here is the G2 to M transition. And I said a little bit about this a second ago. Basically, this is the cell is going to pause here until we're sure that the cell size is big enough to move on. There's no DNA damage, and the chromosomes are totally replicated. Because if there's a problem with this, you're going to end up with two bad cells, and they will either die or become cancerous and cause even more problems. So that's an important restriction point too, to make sure that you don't end up with bad cells in the end. And then there's this mitosis, this metaphase, anaphase one, which just makes sure that all the chromosomes are attached to the mitotic spindle. Because if they're not, then both sister chromatids go to one cell. So one cell ends up lacking, the other ends up with extra, and both cells pretty much die. So that's what that one's all about. So, all right, so mainly we're going to start out talking about these two and how they're controlled. And the early research into this that figured out a lot of this stuff was all focused on this one. And this one's the easiest one to work at because you've got a whole population of cells that are normally at this transition. And those are your oocytes. Xenopus oocytes go through one round of meiosis, actually, not mitosis. And then they stop after one round of meiosis. And when they get stimulated, boom, they go into meiosis too. So if we're looking at oocytes and their transition here, we can do a number of different different experiments. So what I was talking about was here. So you take one cell that's in mitosis and you fuse it with the cell that's in G1, and suddenly that cell that was in G1 goes into mitosis. And you can see it's not even ready to go into mitosis because it's only got one copy of its chromosome. Here you've got two sister chromatids. This has only got one copy. So something in the cytoplasm here of this mitotic cell caused this G1 cell to go into mitosis. Similarly, for the other checkpoint, here's a cell that's already in S phase. You fuse it with the G1 cell, and that other cell goes into S phase as well. So clearly there's something in the cytoplasm that's causing the transition of the cell into the next stage. And that's obvious. Of course, there's something in there that's doing it. It's not magic. But this is the experiment that gives us the opportunity to find out what that thing is. So what you do for the next step then is you take a cell that is in mitosis, suck some of the cytoplasm out, and inject it into a cell that's not in mitosis yet. Boom, that one goes into, well, meiosis in this case. Okay. So what does that tell you? We were able to pull some cytoplasm out that caused the cell to go in meiosis. So there must be some factor in here that's doing this. It's not all the cytoplasm. There's probably like one or two proteins in there that are necessary. So now you take this cytoplasm and you divide it up into all its proteins, and you check each protein to see what protein is going to cause meiosis. And you narrow it down till you find out, oh, look at that. This is the one. This is the one that causes meiosis to start. So, and because meiosis, this is a maturation process, they call it maturation promoting factor, MPF. So all of this is to say, this is how they figured out what the proteins were that caused the transition from G2 into mitosis, by taking the cytoplasm out of one cell and injecting it into another, and then fractionating this cytoplasm until you found out what proteins were involved. And it turns out that one protein, MPF, was actually two proteins bound to each other. One of the proteins is a cyclin, and the other is a CDK. So the cyclin is just a control protein, and it's called cyclin because there are a series of cyclins that are going to increase in concentration at certain parts of the cell cycle and then drop off and then increase and drop off. So they cycle up and down through the cell cycle. So that's why they're called cyclins. CDK then is a kinase. That's what the K stands for. So it's a cyclin-dependent kinase. So this is a kinase that's going to be activated by the cyclin. And then this complex then needs to be phosphorylated. We'll talk about that later. So there are other proteins controlling this. But once it's on, it's going to phosphorylate proteins and turn them on. So that's how this process works. The cyclin builds up in the cycle, binds to the CDK, activates the CDK, phosphorylate target proteins, cause the cell to go through that transition, and then the cyclin breaks down and goes away. Ubiquitination and targeted degradation. So we'll see that in detail in this class. I've been talking about it for months now, but we'll see exactly what that is here. Okay, so you got your two main proteins, your cyclins and your CDKs. And this is how that system kind of works for this G2 to M transition. This is a cyclin B, okay? Cyclin B is really low starting in the middle of mitosis. Right? And you can see in mitosis, once you get through that threshold into mitosis, cyclin B drops off. And then it builds up slowly, slowly, slowly throughout the whole cycle. It's slowly building, 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 and then suddenly it crosses some threshold of cyclin concentration. And then boom, it binds to, it activates that CDK and the activity peaks. So this is MPF activity, which means cyclin came together with CDK, got phosphorylated, and now it's active, it's phosphorylating protein. So this is peak MPF activity. at that peak of MPF activity, you get degradation of cyclin B. And cyclin B gets broken down. It's broken down because it gets ubiquitinated and then degraded. Hang on a sec. I got to pull my notes up because I'm thinking about something I may have skipped over. Okay, so let me just back up a tad and talk more about the cyclins and CDKs. So I said they are regulatory proteins. Let me just talk a little more about them here. Shut up. Seems always yelling at me. All right. Let's make this bigger so we can see it. Way. Okay, so back to our CDKs and cyclins. So our CDKs are the kinases, and they phosphorylate targets that are going to cause those changes seen. And I'll talk about those targets in a minute. And they exist at a constant level in the cell cycle. So CDKs are not changing in concentration. Only the cyclins are. We've got 4 main CDKs. The one for this G2 to M transition we call CDC2. So if you remember back to the first chapter, and I'm sure you do, we saw CDC42. Well, in this chapter, we'll see where the phrase CDC comes from. It's cell division cycle. So again, they were pulling out cytoplasm from cells and like finding what the proteins were. And they're like, all right, we'll call this cell division cycle protein 1, protein 5, protein 42, you know. And so this is CDC2. It's also called CDK1. And then we've got CDK2, which is going to be involved in the transition through the R point. And then CDKs 4 and 6, which will also be involved in that transition point. This will get you up to the R, and this will get you through the R. So those are our 4 main CDKs. And then cyclins are the regulatory proteins. They bind to them, bind to the CDKs, and that causes the activation of the CDKs. Once those CDKs are active, the cyclins will get ubiquitinated and degraded because you want to shut those CDKs off after they've completed their job. And that's the way it happens. And it's usually a feedback cycle. We'll talk about that too. So the concentration of the cyclins rise and fall throughout the cycle. And you've got 4 different isoforms of these. Cyclin B, I just said, is going to get you, I don't know if I said that well. Anyway, cyclin B and CDC2 will get you through that G2 to M transition. And then these 3 will be involved with CDKs 2 and 6. So we'll get to that. I'm such a tease. I'm such a tease. I keep saying, oh, we'll talk about this later. I just want to keep you on the edge of your seats here. I know how exciting this is when you first learn it. Okay, that's all I have here, right? Okay, so I'm going to go a little farther since we still have time left. That's about as far as I usually go in this lecture. But okay, so here we are. So the question now is, my question for you is this cyclin B is rising really, really slowly, right? And I said before, it binds to CDK and activates it, right? But it doesn't do that until it hits this threshold. Why? What's that threshold all about? Because this is where it is actually activating the CDC2 to become active. So why is it that this builds up slowly, but MPF activates quickly? It has to do with a series of phosphorylations that's going to be kind of like, I always like to say it's kind of like you've got one of those dart guns where the dart has got like a suction cup on the end, and you go, push it in until it clicks, and then you pull the trigger and boom, it shoots out. It's kind of like that. It shoots up really fast because of the way this phosphorylation system works. Okay, so here's how it works. You have your mitotic cyclin and your mitotic CDK, and they come together. Now they're not MPF yet because they're not active. Okay, they're an inactive complex of CDK and cyclin. Then you get two inhibiting kinases in, and they phosphorylate in two different sites. So you took an inactive complex and you phosphorylated it to inactivate it. So it's still inactive, okay? So it's been like hyper inactivated. It just is really slowed down. Okay. So now you get another kinase coming in, an activating kinase, and you phosphorylate it again. So now it's got that activating kinase on it, but it still has these inactivating kinases on it. So it's still inactive. Then you have this phosphatase. Now this phosphatase is the rate determining step. If you've had biochemistry, and I hope you have, it's the RDS. This is the slowest step. So as the mitotic cyclin is slowly building up in the cell, it's all going to this form really quickly, very quickly becoming phosphorylated and sitting here inactive. And this phosphatase then is going to take those two inactivating phosphates off, but it's really, really slow. So a little bit squirts through, but not much. But once you get this active MPF, okay, the active complex, it comes back around and stimulates that phosphatase. So now you get more complex being active, more stimulation, more activation, more stimulation. So it becomes this positive feedback cycle that feeds on itself. And so you go from really low to really high fast. As I like to say, it's like the example I always like to use is birth, because I was traumatized by my son's birth. It was really hard. But the way birth is, the uterus stretches and stretches and stretches as the fetus gets bigger, and it gets to the point that the stretch on that uterus causes the release of oxytocin from the brain, which comes down to the uterus and causes it to contract, which then puts more tension on the uterus, which causes more oxytocin release, more tension, more oxytocin, more tension, more oxytocin. You get this positive feedback cycle going until the uterus contracts, and you know what happens after that. So positive feedback cycle. So that's how, this is building up really, really slowly. But once you get some of this to go through and activate it, it does this cycle, and then it, boom, shoots up really fast. Then when it reaches this peak, MPF itself will phosphorylate cyclin B, and then cyclin B will become ubiquitolated. So at the peak of MPF activity, you can see cyclin B dropping off. And so basically this protein is turning itself off. Let me go forward a little bit. Okay, so this is ubiquitolation. So talked about it since way back when. I keep talking about it. Here's a little more detail on it, but I think hopefully you all understand this already. We saw this in ARAD for the first time, ER-associated degradation. So here's a protein, okay? It's going to be targeted usually by phosphorylation. So something phosphorylates this protein, like beta-catenin gets phosphorylated by GSK3 beta, and that tells the cell that this needs to be ubiquitolated. So this protein that's been targeted then gets chains of ubiquitin put on lysine residues. So they only get attached to lysines. And ubiquitin is a small protein. I think it's like 25 or 40 amino acids or something. So a whole series of these get linked up in a chain on it. And those are kind of handles for the cell to grab to get rid of this protein. So this then gets transported over to the proteasome, which is a large protein complex. And the ubiquitins are released and they get recycled to be reused. And the protein gets shoved through this proteasome and gets broken up into short peptides. And those peptides then will be degraded and the amino acids recycled. So targeting that protein basically just has it specifically being degraded. All proteins in cell will eventually be degraded when they become non-functional, but it's not through this process, it's through a different process. This is targeting that protein to specifically break down. Okay, so back to this. When MPF is active, what happens? We've got 4 important phosphorylations that are going to cause everything to happen. So we're right here. So we're in G2, and what's going to happen is we're going to transition into prophase. So that active MPF then will phosphorylate those nuclear lamins and cause the nuclear envelope to break down. They phosphorylate condensin that I said is involved in chromosome condensation. And they phosphorylate proteins, the non-motor maps that cause the mitotic spindle to depolymerize and then repolymerize. So phosphorylations by this are causing all those 3 things that cause the 3 events we see in prophase to happen. And the 4th thing it does then is phosphorylate itself. Okay, so this will phosphorylate itself and then cause the degradation of the cyclin, and that's going to stop MPF activity. Now that's just rude. Oh, you're not done. That's going to stop MPF activity, because once you're at the end of mitosis, you want to stop that and go back into interphase. And so at the peak of activity, it shuts itself off, and then the cell can go to telophase and beyond. Okay, 11:58. That was 45 minutes of lecture. I'm sorry about all the trouble I had. I think I need to take my computer into the shop today. Any questions? Any comments? Oh, we've got a real skeleton crew at this point. All right. Well, I will post this lecture up as soon as the recording saves, and then hopefully you can glean something from that. Okay, everybody good? All right, see you later, guys.